We will start with immunity, and in today's lecture we will discuss about lymphoid organs, T and B lymphocytes, and types of antigens. So what are lymphoid organs? These are basically classified into central lymphoid organs and peripheral lymphoid organs. Central lymphoid organs are thymus and blood cell equivalents. So from bone marrow, the lymphocyte precursors migrate to these lymphoid organs and they undergo processing. Peripheral lymphoid organs are lymph nodes, spleen, tonsils, submucosal aggregation of lymphoid follicles in GIT, pulmonary system, urinary system, and bone marrow. Now about T and B lymphocytes. So only 2% of the lymphocytes, these are in the peripheral blood. Rest 98%, these are in the lymphoid organs. So as you can see in this table, site of processing, it is different in both. In thymus, T lymphocytes, they get processed, and in blood cell equivalents, it is for B lymphocytes. 80% are T lymphocytes, rest 20% are B lymphocytes, and T lymphocytes, they can be subcategorized into cytotoxic T cells, helper T cells, suppressor T cells, memory T cells, whereas B lymphocytes, they can be plasma cells and memory B cells. T lymphocytes, they have a role in cell-mediated immunity, whereas B lymphocytes have a role in humoral immunity. So this is a detailed explanation about T lymphocytes. As you can see here, helper T cells, they express CD4 proteins on their cell membrane. Cytotoxic T cells express CD8 proteins. Suppressor cells, also called as regulatory T cells, and memory T cells. Now what is antigen? It is a substance that stimulates the formation of antibody when enters inside the body of a person. Now the antigenic determinant, it is the epitope. It is a very small area of antigen with specific electrical charge, chemical structure, and configuration. What is paratope? It is a site over the antibody at which the epitope binds. Now a word about isoantigens. These are present in only some members of a species. Okay. For example, blood group antigens present on RBCs of persons which determine the blood group. Now, isoantigens. We talked about these isoantigens. Self-antigens, also called as autologous antigens. These are normally non-antigenic. Okay. So certain self-antigens are present in the closed systems and are not accessible to the immune system of the body. Whenever there is damage to these organs, they get exposed. Also called as sequestered antigens. These are sequestered antigens, a form of self-antigens. What originally self-antigens are, they originate inside the body. Okay, and when they are introduced in somebody else, they can evoke antibody response.